so number one is talking about while there is only one species of Galapagos Island tortoise, there are several subspecies. Larger islands with more wet highlands have less vegetation near the ground. Tortoises there tend to have high domed shells and shorter necks, which restrict upward head movement. They also have shorter limbs. They are the heaviest and largest of the subspecies. Smaller, drier islands are inhabited by tortoises with longer necks and limbs, and with shells that are elevated above the neck, which allow them to browse taller vegetation. Based on the information given, which of the following is a plausible explanation for the ancestry of the tortoise subspecies? So what data do you think is important? Or what observations are important here?
The environment.
What about the environment?
That the turtles or the tortoises have different features based on the different environments.
Good. Different features based on different environments. Hallie?
And like food availability, like if the food is, or like the vegetation is higher up, then they seem to have longer necks.
Okay, good. Right, their features sort of affect what food they eat, right? Okay, so we're going to look for an answer that probably has to do with different adaptations responding to different environments or different challenges. All right, so choice A. The subspecies share a common ancestor whose neck length, shell shape, and leg length were intermediate between the two.
Dr. Zins, we
Yes.
can't see.
Why does that not make sense? Lieutenant?
Because like the characteristics that difference that
Okay, there's not going to be that much divergence with a recent common ancestor. Also, is common ancestry just a hybrid of the other traits? No. Okay, it's not some intermediate phenotype, so A's out. B, the tortoises with shorter legs and necks were most easily preyed on as young animals by the rats that were introduced, so they survived on only a few islands. Delia?
You said we should never choose an answer that doesn't include like information that wasn't included in that.
Good. We do not choose answers that have information that wasn't said in the question. We heard nothing about rats. It's an interesting idea, right, but the question did not talk about it. C, random mutations coupled with the inheritance of acquired characteristics resulted in distinct subspecies. Kaylee?
Can't you not inherit acquired characteristics?
Right, you cannot inherit acquired characteristics. Okay? Excellent. So stuff that you acquire in life, either for some transient amount of time or because of a very specific environmental situation, those aren't inherited. Now, epigenetics gets a little weird there, right? But this question isn't really talking about that. So let's see if D works. Individuals with different adaptations in shell shape and leg length best exploited the food resources and left more surviving offspring. Right? A trait is advantageous. If it's advantageous, they're going to survive. If they survive, they have offspring. That's sort of evolution in a nutshell. All right, so the answer is D. Number 2. Some of you might have seen this before. Okay? This is a very sort of common situation. We have a graph showing oxygen concentration in the water of a lake, right? Divided into daytime and nighttime. Okay, oxygen concentration on the y-axis, time of day on the x-axis. Who can describe what's happening in the graph before we even get to the question? Evan, what's going on?
It kind of goes up first and it goes down as it becomes dark. So as it becomes brighter in the day, the oxygen goes up.
All right.
As it becomes darker, it goes down.
Good. More and more light, oxygen goes up. As soon as the light is taken away, the oxygen levels go down. So what do we think this is involving? What process involves light?
Photosynthesis.
Good.
So D.
All right, you're jumping ahead of the game. I love it. Okay, he's talking about photosynthesis. Okay, so the answers are probably going to have to do with something that happens in photosynthesis. A, the water becomes colder at night and holds more oxygen. Interestingly, the buildup of the warmth throughout the day probably means the water is actually a little warmer, right, when night starts, okay, than right in the morning, okay? Respiration in most organisms increases at night. You breathe heavier in the daytime or when you're sleeping? Who knows? Maybe you do both. Maybe you're a crazy breather when you sleep. All right? But your breathing usually slows down when you sleep. Okay? Things sort of go back to neutral. More organisms are respiring at night than during the day. That's kind of the same answer, just different worded. D, photosynthesis produces more oxygen than is consumed by respiration during the day. All right? D, even if you couldn't remember the mechanisms of photosynthesis, all right, which again is CO2 in, oxygen out. All right, respiration is oxygen in, CO2 out. All right, that answer makes the most sense out of the four that are given. Number 3, you should all remember this. All right, what is this describing that you've done?
Diffusion.
Yeah, the diffusion lab. You've got your dialysis tubing, different things in it, all right? And after a period of time, all right, the container changes. All right, common laboratory investigation involves putting a solution of starch and glucose in a dialysis bag and suspending the bag in a beaker of water. The investigation is aimed at understanding how molecular size affects movement through a membrane. Which of the following best represents the amount of starch, water, and glucose in the bag over the course of the investigation? First thing first, what happens to the bag over time? Allie,
It inflates
why?
because more glucose is diffusing into the
Not glucose. Stuff is diffusing in, but what is it? Where is there more stuff, in the bag or in the cup? Well, if you're thinking of volume, yes, but where is there more solute? The cup just has water. The bag has starch and sugar in it. So water is going to move to where there's more stuff. That's why the bag gets bigger. Water is moving in. Okay? So we need to look for a situation where water, okay, is increasing in the bag. All right? Starch doesn't look like it went anywhere. All right, but glucose is leaking out, right? Because in the second situation here, we have glucose in the cup too. So which graph shows water increasing, glucose decreasing, and starch staying the same? A is correct. Okay? Are there any questions on that one? Like how that works? Again, remember diffusion. Water is going to go to where there's more stuff. Okay, let's skip number 4. Let's go to 5. Which of the following depicts the most probable pathway of processing and packaging a secretory protein within a eukaryotic cell? What does secretory mean? Secreted. What does secreted mean? Yeah, it goes out. So this is a protein that's destined to go away. All right, what is important about a eukaryotic cell? Why do we need to know that? What's the difference between a eukaryotic and a prokaryotic cell? Uh-oh. What's the main difference? Delia?
One of them has a nucleus and the other doesn't.
Good. Okay. Eukaryotic has a nucleus and also has what else?
Organelles.
Yeah, membrane-bound organelles. Okay? So that's important. That makes sense that all the answers can have organelles that are membrane-bound. All right? So those are the 2 details that are important. Okay? So, if we're making protein, where do we start? Where do the proteins first appear? Good, the ER. All right, that's where proteins are made. And it just so happens that only answer choice B starts with the ER. All right, but just to make sure that we're going in the right order, rough ER, Golgi, that's where the proteins are accessorized. All right, secretory vesicles that bud out from the Golgi, and then the cell membrane, right? Because that's where proteins leave the cell. Answer is B. 6, the figure above shows a model of a ligand precursor being cleaved to produce an active ligand that binds to a receptor. So we're talking about the signal, all right, aspect of signal transduction, all right? Which of the following is most likely to reduce the binding of the ligand to its receptor? A change in the cytoskeletal attachment of transmembrane proteins. I mean, you're shaking your head, why?
Well, I just feel like it's D. That's one.
You love to jump ahead to the real answer. What is, why no A?
I just feel like it would really affect it, I guess.
Why?
Because it really only involves like the receptor, I guess. There's really not much you can do with the receptor here. Or,
You're on the right track. All right, we are dealing with the receptor. The receptor is a transmembrane protein, right? But if it was already designed to bind the cytoskeleton, it's probably not going to bind that ligand anyway. Okay? B, the presence of a large amount of the precursor. Is that going to reduce the binding? What's going to happen if you have a lot of materials to make a sandwich? You make a lot of sandwiches, thank you, okay? So if you have a lot of precursor, you're going to have a lot of active ligand, all right? If you have a lot of active ligand, it's going to maximize the binding, right, of the ligand to the receptor. C, an increase in the ratio of the number of unsaturated to saturated fatty acids of membrane lipids. No, that just makes no sense, okay? D, a mutation in the receptor gene that causes a substitution of a charged amino acid for a non-polar amino acid in the binding site. Okay? Yes. Any changes in the binding site of anything, all right, is usually the right answer for a question like this. And why would a charge for a non-polar be important there? What's that going to do? Distance guys have been very quiet. If you have a charged amino acid in the binding site and you switch it for a non-polar one, why would that reduce the binding? Delia.
Maybe because like whatever it's like attaching to has like a negative charge, so then if you change the charge of this one from like charge, well, I guess it doesn't say if it's positive, but
No, that's good. Okay, if the active site has a positive charge, maybe the ligand has a negative charge, and that's why it's able to bind. That's one reason. What's another one, Sarah?
I was just going to say that if you have like a foreign molecule, then you have to have identical molecules so they can interact with each other.
Okay, good. You need those differential interactions. But the protein itself, all right, before it's even folded, right, it folds due to the order of the amino acids and what characteristics they have, whether they're charged or uncharged, polar or unpolar, all right, non-polar, I should say. All right, so if you're switching out charge for non-polar, that's going to completely change the folding profile of that protein. And if you change the folding profile, the active site's not going to look the same as it did before. If the active site doesn't look the same anymore, all right, you're not going to bind that ligand. Okay, so that's why you get reduced binding. 7, cell communication is critical for the function of both unicellular and multicellular eukaryotes. Which of the following is likely true of cell signaling? A, cell signaling uses the highest molecular weight molecules found in living cells. Do we like that answer? Allie says no. Why?
Doesn't really say anything about molecular weight.
Good. It doesn't say anything about molecular weight, and it's not something that really makes sense. Okay? B, cell signaling has largely been replaced No, cell signaling still happens all the time. Similar cell signaling pathways in diverse eukaryotes are evidence of conserved evolutionary processes. Delia shrugs.
I mean, I put that answer, but I don't want
It's funny, again, like AP Bio teachers write these questions. It's almost like they can't help themselves. Like when they're writing the wrong answers, they're like, oh, we're just gonna write something flippant. But then when they want the right answer, it's like very precisely defined. Okay, so C, similar cell signaling pathways in diverse eukaryotes are evidence of conserved evolutionary processes. Okay, again, major themes. You don't actually need to study for that, all right? That's just evolution, okay? Allie?
This has nothing to do with the question.
That's
but
fine.
I realized that most of the right answers are like the longest answer choices.
I wouldn't take that as your golden rule, okay? But I would say the answers that seem like a proper definition of a big idea are usually the answer. Okay, I would say that. Okay, because often these questions, like I've been telling you all year, are trying to get you back to a big idea. Right? They're not trying to trick you. They're not trying to test your knowledge of some minute, ridiculous detail, right, that you would need to memorize. They're trying to say, hey, do you understand what evolution is? Do you understand how energy flows? Do you understand how genetics works? So if there's a definition in the answer that you know is a right definition, that's probably a good choice for your answer. Okay? 8. Which of the following best describes the function of the coenzymes NAD+ and FAD in cellular respiration? This requires you to recognize what these are. This is probably the closest thing you'll get to a recall question, right, on the AP exam. Who remembers what they do? Evan?
Aren't they like carriers, electron carriers?
Yeah, they're electron carriers. Okay, they're high energy electron carriers. So let's find an answer that says they're high energy electron carriers. All right, or at least electron carriers. A, they participate in hydrolysis reactions by accepting protons. Kaylee, is a proton an electron?
No.
Good, your mom will be very happy. Okay. B, they participate directly in the phosphorylation of ADP to ATP. Nope, they don't have phosphate groups. C, they serve as final electron acceptors in the electron transport chain. This one is one you have to keep in mind that might be a little tricky, so we'll keep that one aside. D, they aid vitamins in the breakdown of sugar. That doesn't happen in respiration. Or they accept electrons during oxidation reduction reactions. What's the difference between C and E? Kaylee?
For C it says final electron, but doesn't it like happen throughout the cycle?
It does. Okay, they happens throughout the cycle. They're not the final ones. Right? Those who have had Mr. Psalms, this is like his big thing. They always remember that water is the final electron acceptor, right, in these processes. Okay? But NAD and FAD work throughout the process. So they are just electron carriers. E. Back to chemistry, the diagram below shows energy changes in a specific chemical reaction with and without the addition of an enzyme to that reaction. Okay? And this is a very standard, right, diagram that you've seen before with a catalyst, right? It lowers the activation energy needed to get from reactants to products. Lowers that pump. Which of the following questions can best be answered by the diagram? Again, this question requires almost zero pre-knowledge. You're just looking at it and seeing what you can answer. A, does the addition of an enzyme reduce the activation energy? Okay, that one can be answered by the diagram. B, does the addition of an enzyme result in formation of covalent bonds? That can't be answered. Okay. Does the addition of an enzyme produce a greater amount of products? That can't be answered. Okay. That one could be tricky though, because you see products on the graph, all right, but what is it measuring? Energy and just whether it happens or not. It's not giving you a sense of how much there is. Okay? And then D, does the addition of an enzyme change the pathway? Okay? We don't know anything about the pathway. All right, so the answer is A. Grade yourself so far. How are you doing? Not bad? A few here and there? Okay? Again, these are the style you're going to be exposed to. Okay, so if you're comfortable working through these, which is why we're doing this, you'll be fine in May.